Welcome, FibroFighters. Today we are visiting with Dr. Robert Negerney at his Cancer Institute in Long Beach, California. Last year, we began a dialogue concerning the lack of available options for treating Fibrolamellar and how the Negerney lab could be instrumental in introducing a new way to find drugs for clinicians to consider. Since Fibrolamellar does not currently have a standard of care and there is only one clinical trial, FibroFighters felt it was imperative to find more viable treatment options. Historically, clinicians have been relegated to using standard HCC drugs to treat Fibrolamellar, recommend clinical trials, or take the approach of finding a drug that insurance would approve. Many of these therapies proved ineffective while creating adverse side effects and occasionally causing long-term negative consequences. After many discussions with Dr. Nagourney and his team, we were able to develop a comprehensive Fibrolamellar assay. Dr. Nagourney took on this challenge and believes he will help fill a critical need for our patient community. Since we began this project in March, we are proud to report the completion of 8 successful screenings. We are very enthusiastic about the results being shared with patients and clinicians, offering many new drug possibilities that may not have been recognized without utilizing this form of testing. For your safety and the accuracy of their testing, the Negerney lab is CLIA certified. This means the lab can report their findings to your medical team for further evaluation and possibly use a drug or combination of drugs found to be most effective during their functional profiling testing. We certainly appreciate Dr. Negerney spending his time with us today. We look forward to hearing his viewpoints on the importance of the Fibrolamellar project and his extensive background researching cancer and treating patients. He will then answer questions which you may find useful. Hopefully, after viewing this video, you will have a better understanding of the process and be comfortable sending your live tumor tissue or ascites fluid to the Negerney lab for testing. Thanks for listening, and now let us meet Dr. Robert Negerney.
Well, Tom, thank you very much for the kind introduction. By way of introduction, I am a medical oncologist hematologist, and I am the director and founder of the Nagourney Cancer Institute located in Long Beach, California. My work over the years has focused upon the study of human tumor biology using individual patient tissues. Recognizing that fibrolamellar patients represent a rare but very aggressive tumor that is driven by a characteristic mutational aberrancy, the question arose, how best can we treat these patients? Now we know that the chromosome 19 400 kb deletion that leads to the DNA JB1 PRKACA fusion appears to be characteristic of this cancer, but the question is, what drugs, treatments, combinations, and therapeutic interventions can provide better outcomes? Unfortunately, in this population of generally young patients, conventional chemotherapies and standard drugs are not working very well. When I became interested in cancer biology originally, the field was moving very much toward genomics. Cancer gene informatics is an approach that gives you the lay of the land in cancer from a standpoint of what genes, what mutations, what splice variants, what fusion proteins are present. The one piece of the puzzle that isn't always available is what do we do about it? Simply knowing the roadmap doesn't tell you where the traffic jams are, where the construction sites are, where the accidents are. You need something, another level of depth, another level of interrogation, and that is what we call phenotypic analysis. By way of explanation, your genotype is the genetic material that every cell in your body contains. That genetic information was given to you by your parents. However, what makes you you is how you read and use those genes, and that, your biological features, is known as your phenotype. Phenotype, phenotype. So we became interested in studying not only where you started, but where you wound up. And by doing that, we became interested in how drugs and combinations and targeted agents and signal transduction inhibitors and metabolic inhibitors can cause the process of programmed cell death. One of the reasons that people were not more successful in using human tissue to study cancer biology is that they had conceived of cancer as a disease that was solely driven by cells that grow. Everyone thinks of cancer as unbridled proliferation, the synthesis and use of DNA, and rapid growth of cells. But in fact, most cancers aren't driven by how fast they grow. They're actually driven by how slowly they die. Every cell divides every 2 or 3 months. If the cell divides and the parent dies and the daughter cell carries on, that's the normal homeostasis, ebb and flow of life. However, when cancer cells disrupt that relationship and the parent and daughter stay alive, then 2 to 3 months later, the parent and daughter divide. Where there was 2, there are 4. The next generation, where there was 4, there are 8. And pretty soon, you've got 256 or 512, and the numbers go up and up exponentially, but not by growing too much, by dying too little. Recognizing this, I launched a laboratory model that measures how drugs kill cells, not stop them from growing, kill cells. And when we did that, we changed the way cancer research was being conducted, because we suddenly could, in fact, use each patient's tumor to discern what drugs and combinations would work. Over the years, we have used this technique in very helpful ways. One of my first discoveries was in a leukemia called hairy cell leukemia, and today the treatment that we discovered at Scripps Clinic in La Jolla is the treatment of choice for hairy cell leukemia. We also identified synergies between chemotherapeutics and leukemias that became a standard of care in the treatment of chronic lymphocytic leukemia. We then went on into solid tumors and discovered the first uses of cisplatin gemcitabine doublets, most of which are used around the world today. So when Tom Stockwell came to me and said, "Can you help me with this desperately difficult problem?" I thought, it's a perfect opportunity. Here is disease that we really don't know how to treat. We have at our disposal a number of interesting drugs, some of which are very interesting, but no one knows quite how to use them, and no patient can take every drug. So we need a way to discern their likelihoods of response. We need a way to pick the winners and the losers, to separate the wheat from the chaff without exposing patients to toxicities. Now, I'd love to be able to tell you that every time I pick a drug it works. That's not true. I'd love to be able to tell you that when I pick a drug it isn't toxic. That's not true either. But what we try to say is, if you're going to take a drug, make sure it's got the best chance of working. Now, in our experience, in over 2,500 published peer-reviewed experiences, we have now shown that when we pick a winner, when we pick a drug that looks good for a patient, the chance of their getting better is 2.04-fold, and that's a statistically significant finding at p.001. And the chance of that patient being alive a year later is statistically significant better at 1.44-fold higher survival, and again, statistically p.02. So with that data set, when Tom approached me, I said, yeah, I think we can go after patients with this difficult, aggressive, hard to treat cancer and try to peel back the onion layers of complexity so that we get down to the best treatments. Now, interestingly, we've only done a handful of cases so far, but there are patterns that we are recognizing, some of which identify classes of multi-targeted tyrosine kinase inhibitors that, to my knowledge, have not been directly applied in this disease. So I think even just now, in the very, very beginning of this project, in our very earliest stages of our collaboration, I can say that we're finding interesting discoveries, and I'm hoping we'll find more. It's still early. We don't have a large data set. We haven't been able to really create the kinds of distribution curves and standard errors and standard deviations that give us the best handle on patients. But now, with this newfound opportunity, I think we can have a real impact on this rare and aggressive cancer. And with the population so young and otherwise so healthy, if we can find a good treatment for them, the chance that they're living a good life is going to be improved. And we're immensely excited by this opportunity.
So what is functional profiling, and how can it help someone with a rare cancer such as fibrolamellar?
Functional profiling, as the word would suggest, is function. So we have a lot of tools and techniques to measure static analytes. We can look at genes, we can look at transcriptomes, but we don't have a dynamic process to see how those genes and how those protein structures interact with each other to create the final function of the cell. In order to do that, we need cellular studies. We need how cells talk to each other, their microenvironment, their stroma, the cytokines, the inflammatory cells, and all of the features that make up a tumor. So what we've done is to use a functional tool, the cellular microaggregates or organoids, directly out of the body. And using those gives us a handle right on how cells behave when they're exposed to drugs and combinations.
Excellent, outstanding. And this particular assay, what is unique about the Fibrolamellar assay versus, let's say, just a standard shotgun approach for other cancers?
Well, the platform that we use, the ex vivo, meaning out of the body analysis, of programmed cell death was developed some years earlier. When we apply the same platform, we don't apply the same drugs. So, for example, a leukemia would have a collection of leukemia drugs. Solid tumors like lung or breast would have a completely different collection of drugs. Fibrolamellar tumors have an entirely different collection of drugs, such as the carbonic anhydrase inhibitors, many of the multi-targeted tyrosine kinase inhibitors, some of the metabolic agents that we're testing. So the unique feature of the Fibrolamellar assay is the same platform, but applied to very different classes of drugs, sequences, and combinations.
Fantastic, that's great. And how many years have you been using this method of functional profiling?
I first became interested in the concept of studying human cancers when I was in my training, when I was a resident. I then continued that work during my fellowship and wrote my first papers on selecting drugs at that time for leukemia. As I finished my second fellowship and applied this in very unique ways, I realized that I could actually affect the way patients' outcomes could be predicted. I could actually pick drugs and get winners that no one else could get. When that slowly evolved into my current research institute, I decided that this was going to be how I would treat all my patients. And today, as much as possible, I only treat patients based on their tissue studies. We've been doing this for over 20 years.
That's fantastic. Can the patient's clinician add a specific drug of their choice to check on sensitivity if they have a particular specific thing that they're wanting to target?
We love to interact with the treating physicians. We think it's a terrific opportunity. The only caveat is that when you predict a drug will work or not work, you need a database. You need to say, this drug doesn't work because in the 200 people we've tested it on before, the ones that it worked on and the ones that didn't work on clinically, you fall in the range of the responders or non-responders. Now, if you give me a brand new drug and I have no idea what concentration ranges and testing conditions, then I can only say that it did or didn't kill the cells, but it might be at a concentration that is astronomically high compared to what the patient's blood levels could achieve. Or it may be that we were way too low and we recommended it didn't work when it would work if we knew the concentration range. So what we usually say to doctors, if you want a drug tested and we have a database on it, usually at least 20 studies previously tested, then we will add any drug a doctor wants. If we have never tested the drug before, we discourage that, because we don't know what the answer means, and this is so critical because these are patients.
Right.
And if I say to you, this drug is terrific, and you give it, it doesn't work, that's because of the concentration. We did that once, actually, with a drug called oleandrogen. Oleandrogen is a cardiac glycoside extracted from a plant you see along the highway. That drug is very potent as an anti-cancer drug, except that the concentrations it took to kill cells in the test tube were 1,000 times higher than you could safely get in the blood. So we did the study for the group, and we said, yes, it works. But when we compared it to the blood levels, we said it works, but it's too toxic when you give it. The doses you would have to give, the patient's hearts would stop. So that's what we want to avoid, is recommending a drug because we don't know what it works for. So when a physician wants a drug added, like let's say someone says, I want you to add doxorubicin, or I want you to add carboplatin, or I want you to add gemcitabine. Of course, I have thousands of studies I can look. But if you say, I'd like you to add a new drug no one's ever tested, we're hesitant to do that, only because we can't provide useful information.
That makes perfect sense. Is it true that ascites tissue is every bit as viable as live tumor tissue?
Ascites fluid is live tumor tissue, so long as it's cytologically positive. What we mean by that is when you get a fluid out of the body, you send it to the pathologist and they do what's called cytopathology. Cytopathology is concentrating down the cells, examining their features, their morphology, their nuclear cytoplasmic ratios, their cohesiveness, their staining characteristics, sometimes their immunohistochemistry, and saying, yes, the cells floating around in this liquid are cancer cells. We get them all the time. But when we get a fluid, particularly with fibrolamellar so far, we don't often see cancerous fibrolamellar cells floating around the fluid. More often, the liver injury associated with this disease causes the liver to sweat, as it were, to exude fluid. And these exudative or transudative fluids are not cancerous. So you get liquid, but it doesn't have cancer. Now, we're perfectly happy to process them. But because we do so many of these, I can see immediately if there's cancer in it or not. And so we welcome people to send us fluid, but they have to realize that unless we find cancer, we won't set it up.
Got it. That makes great sense. What is your response to any naysayers who say this offers no real benefit to the patient or their clinician?
We have to remember that the field that I am engaged in began in 1954. The first paper in this field was published in 1954 by investigators who published a very interesting article in the Journal of the National Cancer Institute. What these investigators did was to take slices of cancers, put them into a medium, add drugs, and then measure whether the cells lived or died. Sound familiar?
Yeah, it does.
The trouble is, this was in 1954, and the concept of apoptosis, programmed cell death, and all of the features of cancer that I'm studying were not yet described. So these doctors were using a test almost accidentally that turned out to be very smart, and it worked. But the trouble was that at the time there were only 5 active drugs, most of which we don't use at all, aminopterin and uracil mustard, drugs that we would never use today. So the doctors said, well, it works, but we only have 5 drugs, and we can give everybody all 5 drugs all the time anyway, so who cares? So then the next push for the next few decades was to develop drugs. And by the time the 60s and 70s rolled around, there were lots of drugs. But at that time, people said, well, cancer is a disease of cell growth. Cancers are growing too much. So all of the tests in the 70s and 80s clocked around to use drugs to stop cells from growing. That was a mistake. When they did that, all of the large series of studies were negative. And they didn't go back to the source documents and think all this through. They just said, oh, these tests will never work, we're never doing them again, and the whole field fell flat. And that was just about the time that I was doing my residency. And I said, well, wait a minute, let's go back and think through what led to the error. Like, did we make a right turn when we should have made a left turn? And I became interested in the concept, I didn't even know the term yet, in the concept of cell death as opposed to cell growth. And that worked in leukemia, and then it worked in other tumors, and then I read the 1972 Kurwile and Curry paper from the British Journal of Cancer that described apoptosis. And I suddenly said, that's why it works. That's why cells that die are the ones that respond, and cells that don't die are the ones that don't respond, regardless of whether they grow or not. After all, a cell that dies is dead forever. A cell that doesn't stop growing can grow another day. You know, it's like to fight another day. It lives on to fight another day. So the cancer cell has to be killed outright to be sterilized. And when I made that observation, it wasn't until decades after the publication of the first paper on the topic that I suddenly connected them. And I wrote a book called Outliving Cancer, and there's a whole chapter about my making the connection between what I was doing, that everyone was telling me didn't work, and everyone's new concept of apoptosis. And since then, I've really never taken the naysayers seriously. I don't care what they say, because it works. And we've published thousands of outcomes. We've changed the course, the history of breast cancer management, ovarian cancer management, leukemia management, lung cancer management. So they're just going to have to catch up.
Wonderful. Thank you. Last question for you is, besides the potential for immediate treatment options, what could be some additional long-term benefits for our FLC patients and community as a whole?
The immediate benefit is to the patient finding the best drug. But if you drill down to the next level, the fact that this particular mutational aberrancy, this fusion protein, responds to a given drug or combination begins to tell us how it works, it is a functional interrogation of the pathway. You see, when I tell you that a patient responds to a drug, you can then work backward, reverse engineer. What does this gene mutational aberrancy tell me about a functionality? Because you see, right now we just know you've got this DNA JB1. You've got it. Have a nice day. No, no, no. You need to know the implications of that gene so that it crosstalks to MAP kinase or phosphonosyl kinase, and that's where we have to interrogate not just the presence of the gene, but the functionality. Think about the gene profile like a fingerprint. Think about what I'm doing, like apprehending the criminal.
Mm-hmm. Excellent. Well, we certainly hope you put it in jail. That's certainly the goal.
Or executed.
Yeah, well, that's wonderful. Thank you for taking some time and answering our community's questions. We sure appreciate that. And now, if you don't mind, will you give us a tour of your lab where all this great stuff happens?
Sure.
Wonderful. Thank you.
So we took it over because it's such a and so it was ideal. It's got all the facilities, it's got all the necessary equipment, so it's been perfect for us.
That's awesome. Well, it's close to the hospital, too.
Yeah, right from us.
Okay, so now we're in the lab, and Dr. Negerney is going to show us around, and this is where all the findings happen. So, Dr. Negerney, give us a tour.
All right. This is where we do our work. The wet bench area starts off with the samples arriving, often by overnight courier. Once we receive the sample, it is turned over to the technical staff. Eric here and Steve are working under hoods. These are laminar flow hoods that maintain perfect sterility. Once the sample is received, if it's a fluid sample, it's centrifuged, and the centrifuge is over to the left. If it's a solid tumor, then mechanical and enzymatic disaggregation chops these solid samples down into small fragments. They're washed, resuspended, and examined. If there is a good yield and the samples are of desired size, usually in the range of 30 to 70 cells per cluster, then those are resuspended to a concentration for study, and then we outline drugs and combinations of interest. Now, fortunately, we have a panel that Tom has helped put together, which gives us exactly the drugs we want to test each time. And wherever possible, we test all of them each time. Occasionally, we get more than one sample from a patient, and we try to compare lymph node to solid tumor or fluid to solid tumor. Not every sample works, so when it's a good sample and we can complete it, we'll do, I think currently the panel's 24 drugs and combinations. So we try our best to get those done. Sometimes we can't, so we prioritize them. Working in the background, Eric is in the process right now of processing the sample. Steve, to your left, is basically the guy who runs this laboratory, so he oversees all technical activities, maintains all of our quality controls. He's our CLIA expert. He does all the things that make it possible for us to stay legal and alive. And so he's over here. Steve, you want to just introduce yourself?
Yeah, hi, I'm Steve Evans. I've been working with Dr. Nagourney for 30 plus years, since 1986. We've done, I think, over 10,000 patients assays. It's a great way to find out what will work for someone and also what drugs don't work. And I must say personally, I'm very excited to be working on fibrolamellar disease and hopefully having a big impact on a disease that people don't know enough about to treat. So really honored to be able to work on this. Thanks.
So we're working today. Eric is in the process of working up a case right as we speak. This is a patient from Chicago. I know we had quite a lot of cases from Chicago.
Quite a bit out of Rush University.
We receive samples from all over the world. Rush, Dr. Shade has been a terrific ally, and we're so happy to have his expertise. I've actually referred patients to him with other diagnoses based on the connection that you've allowed us to make. So it's a nice
That's
collaboration.
Wonderful. That's wonderful. And it wasn't that long ago. I think it was maybe a month or 6 weeks ago that we actually got a sample from Sydney, Australia.
Yeah, that's right. I remember.
Which is amazing. So we want to make sure that we're reaching out not just to the U.S.,
sure,
but to people in the U.K., people obviously in Australia, because it's universal. So and again, we just so appreciate the work that you're doing.
That's sure. We're happy to do it. Happy to have the collaboration. So Tom, I thought it would be interesting to just look at one of our presentations. This was from the American Society of Clinical Oncology several years ago. It's about lung cancer, so it's not a fibrolamellar case, but it pretty well tells what we're talking about.
Okay.
So remember, beginning with fresh, healthy cells in the tissue culture environment, drug exposures cause the cells to undergo the process of programmed cell death, and by staining and morphology, you can see that these healthy cells are dead. Using that approach, we took a group of patients with lung cancer and we picked amongst the drugs that were out there. Now, at the time, the response rate for non-small cell lung cancer was 30%. Using this approach, we were able to extend not just to drugs and combinations, but also, as you can see here, signal transduction pathways. Now, this is much more the realm that we're in now with you, because fibrolamellars don't respond to cytotoxic drugs. So we're more interested in the signal pathways and all the small molecule inhibitors. So those are what we're working on, like regorafenib and nivantinib and everolimus.
Right.
Those are all on here as pathways. In any case, with the lung cancers, we took the lung cancer population, gave them exactly the drug that matched them, and we doubled the response. Hands down, doubled the response rate. And we also extended the survival. Patients on this trial, some of them are still alive 12 years later.
Outstanding.
Yeah. So this curve of survival never fell off. We continued patients for, this is a patient in October will be 12 years. She had metastatic non-small cell with brain metastasis and presented with a seizure 12 years ago.
Wow.
She's still alive.
So now what we have to work on is getting a poster board like this with fibrolamellar.
Well, let's hope
see
so.
that exact same type
I
of
love it.
Yeah.
I would love it. So here is an example of programmed cell death, all of the three features of programmed cell death, and how we can apply it for any kind of drug and any kind of disease. Okay?
Thank you.